We're going to be talking about the plasma membrane, which is the same thing as the cell membrane. And that is really big letters. Plasma membrane and cell membrane are the same thing. So there's really no distinction there that we need to worry about. You guys are okay calling the cell membrane the plasma membrane. Do not call them the cell wall. That is a big no-no. If your skin, here's an analogy. If your skin is the plasma membrane, then wearing body armor is a cell wall. Okay? Now, I know that you guys put up your iPhones up behind me, most of them, but it also includes watches. So stay off those as well. Your plasma membrane is going to look like this. So we're going to do a little artsy and crafts, some drawing up here. We're going to label everything, but before I do that, everything today that I color in red is going to be considered a lipid. Everything that I color in blue is going to be a protein. And everything that I'm going to color in green is going to be a carbohydrate. The plasma membrane is made up of these three macromolecules. You're not going to see nucleic acids. Those are down in the nucleus, and they're also in a couple other organelles. So it is color time. It's important to have some colors with you. Life is in living color, so this is life. This is biology. We're definitely going to be using colors. So first thing I'm going to be drawing is the plasma membrane. Or excuse me, a phospholipid bilayer of the plasma membrane. So we have the phospholipids, which are a phosphate head and two fatty acid tails. The next thing I'm going to draw is an integral protein, and I'm just drawing everything now. We're going to explain what all these are later and throughout today's class. I'm going to try to keep it color coordinated. Okay, so far I really only have three things drawn. You see one big blue blob? That is called an integral protein. It goes all the way through the phospholipid bilayer from one side to the other. Then you see another blue blob that only goes halfway through. That's called a peripheral protein. And these will all be broken down and explained throughout today's class. So I'm not just going to say what they are and move on. We are going to break it down. Next, you have some of these antenna, like an antenna on your house or the radio antenna on your car to get XM radio, Sirius satellite radio. You have some of these little antennas are coming off of a protein. Then you have some of these antennas are coming straight from the phospholipids. Notice I'm using green. Lastly, you all know that you get cholesterol in your diet. You have to have cholesterol. You can't have a zero cholesterol diet. There's cholesterol that's embedded in your plasma membrane to help it be fluid. When I say fluid, I don't mean like a liquid pouring out of the faucet. I mean always changing, always moving around. Is that supposed to be red? Yes, that's red. Those are lipids. So this is everything that you see in this picture is what's going to be discussed today. And there are 1, 2, 3, 4, 5, about 6 different structures here that we're going to be looking over. All of these together make the plasma membrane, not just the phospholipid bilayer. If the phospholipid bilayer was the plasma membrane, I would take out the proteins, I would take out the cholesterol, I would take out the green things, which are glycolipids and glycoproteins. It would just be a phospholipid bilayer. The bilayer is part of it. And I'll grant you it's the majority of it, but it's not all of it. So, quick breakdown of just what the plasma membrane is. Also called the cell membrane, no difference there. The plasma membrane maintains the integrity of the cell. And it consists of these three macromolecules. The first ones that we are going to talk about today are going to be lipids. And a few things I want to be sure you guys remember about lipids. Are lipids hydrophobic or hydrophilic? We're talking waxes, fats, steroids, and oils. They're hydrophobic. So does that make them polar or non-polar? They're non-polar. Whenever something is hydrophobic, which means water-fearing, it's non-polar all the way. Non-polar means it doesn't have poles. It doesn't have a positive or negative charge, or even a partial charge. What elements primarily compose lipids? Oh, see, I told you. As we get further away from Unit 1, it's going to get harder and harder to remember, so we need to rehash them every now and then.
CHMO.
Carbon, hydrogen, and oxygen. Which two elements are primarily, like the majority of it by far? Carbon and hydrogen by far. There's a little bit of oxygen, not a lot. Okay? Now, one other thing I want you guys to remember, because this is going to come in handy. The difference between saturated and unsaturated fat. Saturated, what state of matter is it typically? It's typically a solid. Unsaturated is typically a liquid. For our purposes today, we don't really need to worry about animal derivatives or plant derivatives. But I do want to go over this. What does a saturated fat lack in its structure? It lacks a double bond. So if you notice here, I have no double bonds. But over here, there is a double bond. That's going to come in handy a little bit later. I'm going to present to you guys, if we would have done and considered the following today, I'm going to share with you what that would have been. All right, so those are lipids. Here we go. There are two lipids that we need to go over as part of the phospholipid bilayer and the plasma membrane, and that is going to be phospholipids. Nothing new here. We did these in Unit 1, phospholipid. You should know that it is a phosphate head, and is that polar or non-polar? The phosphate component of a phospholipid. And you need to, I'm not going to bother saying polar, hydrophilic, non-polar, hydrophobic. You need to know that those two go together. If something is polar, it's hydrophilic. It mixes with water. Water is called the universal solvent because it practically breaks down anything else that is polar. Things that are non-polar are hydrophobic. So the phosphate head is polar, and the two fatty acid tails are non-polar. That is a bunch of gibberish. Can't make that out. Okay. When you get two layers of them, mirror images of each other, they're going to compose the phospholipid bilayer. Bi means two. And when you look at the phospholipid bilayer, as you guys have drawn it just above, what is facing outward, both outside the cell and towards the inside of the cell? And if you're unfamiliar with where this plasma membrane is located, if this circle is the cell. That red line is the plasma membrane. If you were to zoom in on it, you get the drawing that you all have in your notes. So my question is, which way are the phosphate heads pointing? Are they pointing towards the inside of the membrane, or are they pointing outside of the membrane? They're pointing towards the outside. Now, what is the polarity of those phosphate heads? Polar or non-polar? They're polar, so let's put 2 and 2 together. Do you think these polar heads are wanting to face a polar region, both out and inside of the cell, or are they wanting to face a non-polar region? Exactly. The majority of an organism's body on this planet is water-based. The inside of your cell is cytoplasm. You may have heard that word before when you were in honors bio. That's water-based. And then if you go to the outside of the cell, it's called the extracellular space. The space beyond the cell, that's water-based too. And when you hear water, you think polar. Water and polar are hand in hand. So this phospholipid bilayer acts as a barrier. Now, let's continue with the chemistry of this this phospholipid bilayer. We've already talked about the polar heads facing this extracellular space and the cytoplasm. But what about those lipids in between? They don't want to have anything to do with water, which is why they're facing each other on the inside. So here's my next question. Those lipids are non-polar. Water is polar. Would you expect water to be able to simply diffuse right through the membrane from one side to the other? Absolutely not. That shouldn't happen. So cells do run the risk of bursting if too much water comes in. It's called lysis, or they just lyse, or they could shrivel up and be totally dehydrated if all the water escapes. You want to prevent that. So that's what this membrane tries to do. So only small, non-polar molecules can diffuse through. And normally I would follow up that question with a why, but you already answered that. The reason why is because the polarities are different. And I want to remind you that for this class, for my tests, for the semester exam, for the AP exam, you can't just have regurgitate these notes. You can't say, the phospholipids compose the phospholipid bilayer. Only small nonpolar molecules can diffuse through. That's a robot can do that. Anybody can memorize, but you have to know why. You have to know why things are what they are. So when I pose a statement like only small nonpolar molecules can diffuse through, you need to know why. The reason why is because the fats, which are on the inside of the membrane, as you see here, they are nonpolar. Water is polar. Polar and nonpolar do not mix. So if water tried to get into the middle of the membrane, it just wouldn't jive and it can't interact. If you were to go home and you find some extra virgin olive oil that might be in your pantry, pour it into a glass and then fill up that glass with water too. The oil will be on top of the water. Water is more dense, so it goes to the bottom, but they won't mix. You can put them in a blender, they won't mix. Because oil is nonpolar, the water is polar. They just don't interact with each other. It could be vegetable oil, it could be motor oil. Now, moving on. Phospholipids are not just stuck in place. And I want you to use your imagination. I want you to think of a pool. And I want you to think of the pool surface, the water surface, covered in flowers. And the flowers are just floating on the pool. Are those flowers going to stay in one spot, or are they going to move around the pool? They're going to move around. Well, that's exactly how the plasma membrane is. Like if I was to focus on just this phospholipid right here, guys, this one I'm highlighting in yellow, that phospholipid in a few seconds could be way over here. Or it could be over here. Or maybe right there. It doesn't stay put. They move around past each other. It's like students going past each other in the hallways. You guys are moving past each other. You're finding new places to be. It is not like what you are right now. You all are sitting in one spot. You're not moving. That is not what a plasma membrane is like. This big protein right here, it could be over here in a minute. It's always moving around. So we call that like a fluid situation. When we were trying to figure out how school is going to be working this year, are we going in these buildings? Are we doing e-learning? What the heck is going on? Like, it's different every day. No one knew what was happening. It was a very fluid situation. Fluid means ever changing in this case, not like a liquid. So this is a fluid situation. The plasma membrane is a fluid situation. It's always moving around. So it must maintain its fluidity. In order to maintain the membrane's fluidity, its ability to always be in motion, like the flowers on top of a pool, you have to have a proper ratio of saturated fatty acids or saturated fatty acid tails and unsaturated fatty acid tails. Remember that in a phospholipid, there are 2 fatty acid tails per molecule. They can be saturated or unsaturated. Typically, it's one of each. This whole idea of the membrane being able to move around like flowers on top of a pool. You could have flowers and beach balls and a dead frog and this one spider and your grandma floating on a little floaty. Everything's moving around. We would call that the fluid mosaic model. A mosaic is a lot of things. Like if you go out to your pool and you got flowers and a dead frog and some beach balls and your grandma all floating around, that's a mosaic. There's a lot happening. In the plasma membrane, there's a lot happening. There's phospholipids, there's proteins, there's glycoproteins, glycolipids. I know you don't know what they are yet, but you will. They're all moving around, so we have this fluid, which means ever-changing, mosaic, which means lots of stuff, model. So lots of stuff is moving around. The component of the plasma membrane that allows for the fluid mosaic model to actually hold true is our next lipid we're going to talk about, and that would be cholesterol. Now, you may hear that cholesterol is bad for you. It can clog up your blood vessels, and that is absolutely true. But everything in moderation. A milkshake is fine. 12 of them is not fine. 3 eggs for breakfast, fine. 60 eggs, you're pushing it. So there is such thing as too much, but you do have to have cholesterol. One of the purposes or functions of cholesterol in your body is to be part of your plasma membrane. These big red chunks that I drew in the plasma membrane, like that right there, that's a cholesterol molecule. The cholesterol molecule helps maintain the fluidity of the plasma membrane. If you were to somehow, some way take out the cholesterol from the plasma membrane, the membrane would be more rigid, not as flexible, not as durable, less fluid, not very conducive for an ever-moving, ever-changing cell. It would just be very static. Static means not much change. Okay, and by the way, this big blob right here that I drew. That would be a cholesterol. So you do have to have cholesterol in your diet. This is one of the purposes that it serves. But just know, I thought cholesterol was bad. In case you think that way, you guys have to have sugar in your diet. You have to have water in your diet. You have to have all that stuff, but there is such thing as too much. Too much water will kill you. Too much salt will kill you. Too much oxygen will kill you. So we're like Goldilocks. You got to be within the safe range. Too much, too little. Now, while we're on this topic of fats, you do not need to write this down. Just pay attention. If we would have done a consider the following today, here's what it would be. You have two different environments. You have a hot environment and you have a cold environment. Okay? Let's talk about the hot environment first. You go to the bottom of the ocean and there's a hydrothermal vent, which is basically an underwater volcano. It's where life is believed to have began on Earth, at these vents. Well, these vents are spewing out lava. They spew out incredibly hot fumes that would melt you or I. But if you go down to these vents, there's just little crabs. And this crab doesn't melt. So here's what I'm going to ask you. Would you expect the fatty acids, and listen carefully, would you expect the fatty acids that make up his phospholipids, which makes up his plasma membrane, to be more saturated fat or unsaturated fat in order to resist the incredible heat at the bottom of the ocean near these vents? Ryan? You think they would be saturated, okay? Can you justify your answer?
Because of, like, they're solid, so it'd be a little harder, I guess, to melt than being liquid form, which would just, like, evaporate next to the vent, and all of your liquid would turn to gas.
You're right. Yeah. If you could coherently put that into some sentences to justify your answer, yeah, that's right. So here's the cliff notes of what he said. Saturated fats are solid. This creature's in a very hot environment where it like literally risks melting. Like this crab would be crab soup. So to resist liquefying, which is going from solid to liquid, this crab has a chemical structure built into it that is solid, that resists being liquefied. And that would be saturated fat. Are there any questions about how that would work? So let's do the opposite of that. This other crab is on the bottom of the ocean, like near where the Titanic sank, in the Arctic Circle, or near the Arctic Circle. It's at the bottom of the ocean, so there's no sunlight, and it is super cold. But this crab doesn't freeze solid. It's fine. You or I would turn into like a literal ice cube, like Leonardo DiCaprio in Titanic. I'll never let go, Jack. I'm going to let you go now. Why doesn't this crab freeze? Yeah. It's mostly unsaturated fat. What state of matter are unsaturated fats? It's liquid, so it would resist freezing. Freezing is another word for solidification. When you freeze, you go from liquid to solid. You're solidifying. If you have a chemical structure built into you where you resist solidifying and you're less dense, you're going to have a leg up on not freezing to death when you're in these very cold temperatures. So this would have been our CT effort today if we would have done one. It would have been that very problem. And I got that from the 2018 AP Bio exam. So, or at least a question similar to that. But you see how that question, you have to go all the way back to Unit 1 on an FRQ. So don't ever think that Unit 1 is just, whatever. It will be coming back. All right, let's move on. Proteins. Make sure you spell proteins right. It's E before I, not I before E. Sometimes I see that. You don't get counted for misspelling things, but it doesn't hurt to spell them right. All right, so in the plasma membrane, we have these proteins called integral proteins. Emphasis on the in, which means they're inside. This means any protein that is embedded in the plasma membrane. It could be fully embedded into the plasma membrane, like peeking out both sides, or it could just be in one half of it. One membrane that's going to be going all the way through the membrane from one side to the other, like this big blue blob I have right there, that is going to be called a transmembrane protein. Trans means, in this case, all the way through, like transportation, you're moving around. This is going all the way through. This protein travels through both sides of the plasma membrane. It looks like this if you were just looking at it from the outside. It looks like a little blue jelly bean. But this jelly bean has a hole going right through it. Imagine a grape, and you were able to take a little drill bit, like for screwing in screws into the wall, and you screwed a hole all the way through that grape. You cut the grape in half, and here's what you would see. This little channel or tunnel is what allows large molecules to go from one side of the membrane to the other. And so this specific example is known as a channel protein. This is like a tunnel going through a mountain. This allows for molecules that are not small and not nonpolar to pass through. So generally, when you have the phospholipid bilayer just by itself, small nonpolar molecules can go through, but large polar molecules cannot. So they need another way to get through, and that's going to be through these channel proteins. These channel proteins are basically there to let the big molecules and the polar molecules get through. Yes, Jasmine? What's that? All right. Now, what molecule is not element, but what molecule is essential for life on this planet? It's the majority of the surface of this planet. Water. Cells need water too. But hold on. Can water just willy-nilly pass through the phospholipid bilayer? Absolutely not. The chemistry doesn't add up. So you have to have another type of channel that is specifically meant for water. The name of this channel is usually very easy for y'all to remember. It's called an aquaporin. It literally means water hole. You know, aqua is water, pore, hole, like pores on your face. This is a protein that allows for the passage of water. Cells have to have water. Sometimes cells have too much water, and they have to pump it out. Sometimes cells need water, you got to pump it in. Then lastly, the next protein I'm going to mention, if none of you remember the name of it from Honors Bio, that's fine. You'll learn it next unit. But I'm just going to throw it out there, kind of plant that seed for a few weeks from now. ATP synthase. Now, the fact that it ends with ASE, what does that mean? It's an enzyme. All enzymes are proteins. So this fits. It's a protein. It transports hydrogen ions, which are H+, hydrogen atoms with a charge, across a membrane to synthesize ATP. ATP is the cell energy that is made by the mitochondria in the chloroplast. And the protein that actually makes the ATP is called an ATP synthase. And enzymes are named after what they do. So if something's called a synthase, it synthesizes something. In this case, it synthesizes ATP, which is adenosine triphosphate, which is cell energy. When you hear the mitochondria is the powerhouse of the cell, it makes ATP. ATP is energy. All right, so if we talked about transmembrane, well, now we got to get to the opposite. Peripheral proteins. You know, a lot of you are driving now, and you might be at a red light, and the light turns green. And one thing I would recommend all of you do, don't take for granted that everyone else knows that they are supposed to stop and you are allowed to go. Look both ways before you go. Let's say Hannah's driving, and she sees something in her peripheral vision. She's looking this way. She sees it. She stops. Somebody just ran a red light. She saved her own life because of her peripheral vision. Peripheral means your sides. Well, peripheral proteins are on the side. They don't go through the membrane. They're embedded in the membrane, but they don't go through it. So they're just on one side. So if you go back to the drawing at the very top, this guy here is a peripheral protein. Notice that it's on the side of the membrane, not going all the way through it. So these are non-embedded proteins that travel across the membrane. And in a moment, you're going to learn what a glycoprotein is. So I'm just going to kind of get ahead of myself a little bit. When I mention glycoprotein in just a minute or so, whenever you hear the prefix glyc, like glucose or glycogen, that means sugar. But the protein that I'm referencing when I get to glycoproteins in a moment is referring to this guy, the peripheral protein. And you'll see what I mean, hopefully. They basically serve as the base of an antenna for your cell to talk to other cells. Okay, and the last macromolecule we need to talk about are carbohydrates. If you look at the original drawing we put at the top of these notes, there were these little green antenna-looking structures that I had you draw. That's what we're going to talk about next. So what I'm going to do is I'm going to actually redraw 1/2 of a phospholipid bilayer. And I'm going to put a little peripheral protein right here. And I'll just continue the plasma membrane over here. Okay. So, guys, these carbohydrates that we're going to be talking about today, they're not a monosaccharide, which is just one unit of sugar. They're not a disaccharide, which is just 2 units of sugar. And they are not a polysaccharide, which is like thousands or hundreds of units of sugar. We call them oligosaccharides. Now, you should know what saccharide means. What does it mean? It means sugar. Oligo, what the heck is that? It means short. More than the disaccharide, but less than a polysaccharide. So if you really wanted to know the hierarchy of sugars, it goes monosaccharide, disaccharide, oligosaccharide, polysaccharide. Monosaccharide is 1 sugar. Disaccharide is 2 sugars. Polysaccharide is like many, but when I say many, I mean hundreds or thousands. Oligosaccharides is like 10, 12, More than di, far less than poly. So the first one I'm going to draw is going to be coming out of this protein. So let's see if you can put some brain power into this. You tell me, what type of glyco sugar is coming out of a protein? You ready? Here's your choices. Glycoprotein or glycolipid? That's all there is to it. The sugar that is coming out of a protein is called a glycoprotein. And then what would you call the sugar that is coming straight out of the phospholipids? Exactly, glycolipid. So let's define what these two are. A glycoprotein is a short sugar chain attached to a protein. A glycolipid is a short sugar chain attached to a lipid. In this case, it's going to be a phospholipid. What do these two do? They both act as receptors. They receive, I don't think I spelled receive right. They receive chemical messages. You guys are teenagers. You have hormones that are being secreted a lot. This is how you're growing up from a child to an adult. And there's a lot that's entailed in that. You've had these signals your whole life, but they just started turning on when you were like 11, 12, 13, 14 years old. And then when you get to be probably early 20s, they stop, really. You know, at least the frequency of them. Well, for the fellas, let's talk about the guys for a second. You guys had testosterone. When you were in 6th grade, you were these little skinny, light boys, little boys. And then around 7th, 8th, 9th grade, everything just started to change. Started getting a little bit of facial hair. Muscles started to get a little bigger. You started to get taller. Your bones were growing. Voice got deeper. That was the effect of testosterone that it has on various cells of your body. Your cells didn't start to make those changes until they received the signal to do so. When you were 5 years old, you had testosterone. When you're 15 years old, you had testosterone. It was only when your cells received those signals to change is when they actually changed. So it's because of these glycoproteins and these glycolipids that our body is able to communicate with other areas of our body. When you have a big scab on your knee or on your elbow, how is it that your body knows when to stop healing? Why isn't it just keep growing skin and skin and skin and skin and skin? Because it's communicating. Your whole body communicates with every other part of you. And there's really two parts of you, or two different types of receptors. You can take a break now. I just want you to watch and listen. You guys have cells, and you have about 260 different types of cells. Well, let's say that this cell right here is a cell from your bladder. This is who do I want to pick on? This is a Mooney bladder cell. Now, on this cell, there are two different types of receptors. These orange receptors are basically saying, I belong to Turner. I am Turner cell. Hear me roar. It's actually a bladder cell, so it's I am Turner bladder, hear me pee. Now, these purple receptors say, I am Turner's bladder cell. So the orange is Ian Turner. I have your name right, correct? Good. I'm getting better on the names. And the purple one is I am Turner's bladder. So throughout your entire life, you have these white blood cells called leukocytes and lymphocytes. They're going around trying to find anything that's not supposed to be in your body. Anything at all, whether it be a wood splinter, a virus, a bacteria, maybe someone else's cells. And the white blood cell is going to march up to this cell. And they're going to say, I need to see some form of identification. You know, you're down here in the bladder. Prove you are who you are. So this bladder cell says, here's my orange name card. I am Turner's cell. I belong here. I am part of him. White blood cell goes, okay, I see that. You belong in this body. Now, you belong in this part of the body. Prove to me that you're a bladder cell and not some nerdy brain cell or a heart cell. So it shows the purple one, says, I am part of Turner's bladder. White blood cell goes, okay, we're good. Enjoy your day. And this happens all the time. But every so often, you actually get cells of one body part that grow in a different body part. So let's say that they're right next to his bladder cell is a liver cell. It's called the hepatocyte. And this has the orange projections that say, I belong to Turner. But it has these blue projections to say, I belong to Turner's liver. And so the white blood cell is going to approach this cell and say, prove to me that you belong in this body. It can prove that. Okay, prove to me that you belong in this part of the body. I'm in the wrong place. And so what the white blood cells tell this cell that's in the wrong spot to do, basically puts its hand on its shoulder and say, I need you to die now. And it does. This is called apoptosis. Yeah, it's like watching Office Space, like, yeah, I need you to die. And that's what they actually do. The cell will self-destruct if it's a normal functioning cell. The white blood cell tells it to destroy itself, and it does. Now, what if he gets into a terrible car accident and he has the steering wheel just jabbing into his bladder and it damages the bladder so badly the bladder is no longer viable. We have to transplant it. So he needs a donor bladder. You hear about all this talk about a perfect match and like a 5 out of 6 match and all this. Well, let's say that Hannah has a bladder. You know, Hannah's like, I don't need a bladder, I'm good. Just hypothetically, we only have one bladder, so this wouldn't work. But you typically get bladders from people that die in a car accident. I don't want to say, so Hannah died today, and she's an organ donor. We're not going to go there. But let's just say that Hannah she was born with 2 bladders. And so she's like, here, have one of my bladders, Turner. It's okay. And Hannah is a match. She's like the best match you could get. But it's still Hannah's cells, not Turner's. And so when we put this inside of Turner's body, his white blood cell is going to say, prove to me that you belong in this body. And instead of saying Turner's cells, it says Hannah's cells. And despite her best intentions to save her buddy's life, his immune system is going to say, you got to die. Well, what we do is we actually give patients that are transplant recipients immunosuppressive drugs. You're basically stupefying your immune system and making it very weak so it can't fight back. And so hold on, give me 10 seconds. So when his immune system says, are you supposed to be in this body? The cell goes, not really. And the immune system goes, all right, whatever, we're good, you can stay. Because it's weakened. And people on immunosuppressive drugs have to be on those drugs their whole life. So before we all started wearing masks, if you went to Publix or the bus stop and somebody was wearing a mask, it's because their immune system is so weak they can't risk getting sick because they probably had an organ transplant. All right. This will be on YouTube.